All right, so welcome to our first video presentation. It's March 18th, and we are going to talk about RNA today. I know, right? So let's take a look at what we already do know. We've talked about DNA, and you should be able to fill in the left side of this table. So DNA has what type of sugar? And we know it's a sugar, so it ends in OSE, and that would be deoxyribose. Okay, so DNA, how many strands? And that is double stranded, so there's two. And what are the nitrogen bases for DNA? Right, adenine, cytosine, guanine, and thymine. All right, so now we're going to talk about RNA and how that differs from DNA. If you recall our history, Phoebus Levene discovered both types of sugar in the nucleic acids, and he actually discovered the sugar from RNA first. 20 years later, discovered deoxyribose. So the sugar for RNA is in the name, ribonucleic acid. It is ribose sugar. Right. RNA, whereas DNA is double-stranded, RNA is usually single-stranded. And in terms of nitrogen bases, they're not quite the same. Whereas DNA has adenine, cytosine, guanine, and thymine, RNA includes adenine, cytosine, guanine, and uracil. So in RNA, uracil replaces thymine, which means if you were looking at an RNA sequence, there would be no T's. There would be U's in place of those. So if we look at the nucleotides, DNA nucleotide looks like that. We've got our phosphate group connected to a deoxyribose sugar bonded to a nitrogen base. RNA is very similar. Okay, RNA is the phosphate group bonded to a ribose sugar. It's slightly different. And we're not going to go into the details of exactly what's different about it, but just know it's a different sugar. And then that's bonded to a nitrogen base. Right? So all of this information, you should kind of expect that I'm going to be asking you. Certainly the stuff from the table, and just looking at the similarities, differences between DNA and RNA, and then what the nucleotides look like. So looking at this in a slightly different way. So we've got our deoxyribose in the DNA. Double-stranded, friendly little DNA strand there, whereas RNA is going to be single-stranded. It's only half. Ribose sugar, and you've got the bases below. So RNA really is the other nucleic acid. When people talk about nucleic acids, I think just about everybody knows about DNA. RNA is not as well known, certainly not as popular. And it's even by scientists in the past, RNA was really kind of seen as the DNA's sidekick. Whereas DNA was big and important and now had all these superpowers, RNA was kind of sad. And they really didn't think it did much of anything other than just copy instructions from DNA, take it out to where the proteins were made. It was a messenger, and that was about it. We now know that RNA is, far more important. It does way more than what was ever suspected. It now has lots of different roles that we know of in coding, decoding, regulating genes, expressing genes. There are types of RNA that can silence the DNA. So you might have a gene for a particular trait, but if you have silencing RNA, it can actually shut that down and change your expression. So RNA turns out to be way more powerful than it was once thought to be. So types of RNA, and the types you really need to know are these first three. We're going to show you that there's some other types, but the ones that I want you to know are these three. particularly the first two. So the first is messenger RNA, written as a small m RNA, and this copies the genetic code, there's the nucleotides, from the DNA strand, and then takes it out to the ribosomes. Okay. Second type is transfer RNA, abbreviated as tRNA, and this transfers amino acids to the ribosomes and adds them together to make the protein chain. And all of this stuff we'll see in a simulation in a few minutes. The final type is ribosomal RNA, rRNA, makes up part of the ribosome, puts amino acids together, forms the proteins. Most of the RNA found in your cells is ribosomal RNA. Okay, the other types, and like I said, I'm not going to ask you specifically about these, but know that there are other kinds. Small nuclear RNA functions in the nucleus, and it processes the messenger RNA. MicroRNAs, really small, short RNA segments, still kind of figuring out what a lot of them do, because there's many different kinds. Small interfering RNA is a type of microRNA, and it controls gene expression, protects cells from viral attack. We've talked about viruses. Viruses come in, you got to have a way of shutting them down so that they can't, take over the cell once they get there. And you do have things like small interfering RNA that can get in there, damage, alter the viral DNA so that it doesn't affect your cells. All right, so the process we're talking about is gene expression or protein synthesis. And this is the use of information in the DNA to direct the production of proteins. In other words, you're using the DNA instructions to make the actual proteins, which is what it's there for. And there's two stages of this process. First is called transcription. Second is called translation. And transcription, if you think of the word, transcribe means to copy or write down. Translate, if you think of translation as far as how we use it in something like languages, translate just means changing it from one language to another. And really in gene expression or protein synthesis, you're doing the same thing. So if we look at this kind of all at once, you've got the DNA, and there's your DNA sequence and code. When transcription takes place, that is forming the RNA copy of the DNA code, which then goes out through the nuclear membrane and then is translated at the ribosomes to make the protein. So this is a really nice summary of kind of what's going on here. All right, so transcription is construction of messenger RNA along portions of the DNA molecule. Each group of three bases, so you have three nitrogen bases in a row, that is called a codon. And we're going to be talking a lot about codons when we talk about this process. One codon is the same as one amino acid. We have about 10,000 plus genes. Each gene is many, 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 many codons. And so as a result, we have lots of DNA. We have about 3 billion base pairs of DNA. So transcription, let's see this. And we're going to open this up. All right, so here's transcription. And you can see the URL on this if you want to go to this. It's just stolaf.edu transcription. You can play around with this, but basically here it is. We've got our DNA strand, double-stranded. Okay. There is a promoter that is telling the enzyme RNA polymerase where to start. So RNA polymerase is an enzyme. It unzips the DNA. As it does that, the messenger RNA binds to the exposed DNA nucleotides, the bases, and makes a copy. Okay. So this copy of messenger RNA is going to be the same as this side of the DNA strand, except, of course, where DNA has T, RNA does not have thymine, and U will replace it. U bonds to A. And so they hit a terminator, which tells the enzyme, okay, stop making this. And we now have our messenger RNA strand, which is complementary to this half of the DNA strand. Okay, and so that's what we want. Okay, so that's transcription. Translation is the process in which the messenger RNA code is read and converted into a specific amino acid sequence in a protein chain. So the messenger RNA, that little strand we just ended with, goes out through the nuclear membrane, goes out to the ribosomes, and the ribosomes read that code and turn it into amino acids in a protein. And so this is where the translation comes into effect. You're changing this up to this point. You have A's, C's, G's, and T's. That's nucleic acid language. All right. So A's and C's, G's and T's, nucleotides, that's nucleic acid language. They're turning it into amino acids and proteins. That is the protein language. So we really are translating between two different languages. Okay. So amino acids are linked, form the protein, and let's see the translation. Okay, same source, same site, except now we're looking at translation. So here's our messenger RNA strand. It goes out to the ribosomes, and let's get our ribosomes in there. So there's the ribosome. This is transfer RNA, and the AUG on our messenger RNA results in an MET, which is methionine. So that is a particular amino acid. Every time you had an AUG down here, you would always get methionine. But our next one is AAG, so we're going to get a different amino acid for that. That ends up giving us lysine. Okay. So bonds these together, shifts over. We're going to bring in a new one. Okay, and this one is phenylalanine. Okay, so we add them, shift it over, get rid of the end one, and add a new one. And this is pretty much what it does. And it does this very quickly. We're doing this really slow. In real life, this happens very fast. Okay, we have a release factor, which tells it to, okay, you're done, let go. And we've got our protein here, release factor here. This protein is for amino acids. No protein is for amino acids. Most of the proteins we look at are hundreds of amino acids long. This just does it really nice and quick. Normally this would be much, much, much longer. Okay. All right. Yes, it is. So you can try these processes. Learn Genetics is a very good site for this. I will put a link to this on the web page. So now you've got some notes on this. Take a look at those. Tomorrow I will have some practice for you on this. One will be this site. There will be some other sheets on the web page. Check those out. And we'll work on this through the end of the week. When we get back after break, you could probably expect that we will have at least a short quiz on this, and it will be on, particularly RNA. Okay. All right. So thanks a lot. And if you have questions, definitely email me, and good luck.